everyone, my name's Ada Hagan. I'm a graduate student at the University of Michigan in the Department of Microbiology and Immunology. What we're going to talk about today is the battle for iron. What's all the fuss about? So, many people realize that iron is an essential part of their diets. In fact, the character Popeye, his job was to convince us to eat more spinach because of the amount of iron that is supposedly contained. But what many people don't realize is that humans aren't the only ones who need iron. In fact, bacteria do also, and that's what we're going to talk about today. But first, there are a lot of reasons that our cells need iron. For instance, our red blood cells that carry oxygen to the other parts of our body, well, the iron is an important component of that process. In addition, our cells need iron in order to replicate DNA and create energy within the cell. But what happens after you eat these iron-laden foods like spinach or red meat? There are cells that line the gut in your stomach. And the job of these cells is to take the iron as it's digested from the food you've eaten and bring it inside. So once the iron has been brought into these cells, they have two choices. They can either store the iron for use in the future, or they can send it into your bloodstream so that it can travel to the cells that will need it later. Iron is a little bit tricky though, because you actually never want to leave iron alone. It's a little bit like a toddler in that respect. And so this is the job of transferrin. His job is to essentially chaperone this iron through the bloodstream. That way it doesn't cause problems. When another cell needs iron from transferrin, it will put out a little flag, take the iron from transferrin, bring it inside, and use it for the cell processes that it needs it for. But like I mentioned earlier, humans aren't the only ones who need iron. It turns out bacteria do also. And they really need it for the same reasons that we do. Again, replicating DNA, creating energy, and maybe even helping them protect themselves from outside stresses. So if a bacterium is getting all of the nutrients it needs, like sugars and including iron, then they can replicate very quickly. And if we're talking about a bacterium that will cause infections, such as a pathogen, then that quick replication can become a problem. Because transferrin does such a great job at making sure iron is never left alone, however, this means that it can be difficult for the bacteria that are trying to invade your body to access iron. So they have another way to try and get iron. This is through a siderophore. Siderophores are essentially little mini magnets, and the bacteria can send these out into their environment when they don't have enough iron. And the siderophore will find sources of iron and take the iron back to the cell. Now, if we are talking about a bacterium that is in your body, then one possible source of iron is transferrin. So siderophores are generally essentially stronger magnets than transferrin is, so they can pretty easily take it away. And then the siderophore can return to the bacterial cell with the iron payload, and the bacteria can continue to replicate. Obviously, this isn't a good thing for our own cells and our own body, because we don't really want bacteria replicating willy-nilly within, right? These cause infections. So our immune system has a really cool response known as Asadero. So when your white blood cells recognize that there's a bacterium invading and that it needs iron, then they will release the Asadero. And this is essentially a catcher's mitt for siderophores. They can stop siderophores from returning to the bacterial cell by binding to them. And if the siderophores never make it back to the cell with their iron, then the bacteria can't replicate. I know many of you have probably heard of something called the microbiome. This is the collection of bacteria that live in your gut or on your skin or in your mouth. And these bacteria are commonly considered good. Well, as it turns out, most of the bacteria that live in your microbiome make the siderophores I just told you about. Pathogens, however, make a different type of siderophore known as a stealth siderophore. We call these stealth siderophores because they actually can't be bound by siderokalin. Sometimes they have extra attachments that make them too large. Sometimes they're just a different shape entirely, and so siderokalin can't recognize them. Because our bodies haven't created a counter to these stealth siderophores, then my job as a basic researcher is to learn more about what I call the life cycle of a siderophore so that we can try to develop new drugs or antibiotics. Let's walk through the Asadero life cycle together. First, the siderophore is assembled from a number of smaller components within the bacterial cell. Then, it has to be sent outside the cell in a very specific way. Outside the cell, it then goes on to find iron within its environment. Now, depending on the type of siderophore, because there are hundreds of them, and where the bacterium is living, then the source of the iron can be different. Once the siderophore has gathered the iron from the environment, then it has to be recognized by the bacterial cell so that it can then be brought into the cell and the iron removed from the siderophore so that it can be used by the cell for growth or replicating DNA. Now, as you can see, this is a pretty complex process, but with a complex process means we have that many more opportunities in order to interrupt it. So what are some ways we can interrupt the siderophore life cycle? One is to create an antibiotic that stops the siderophore from being made in the first place. The process of making a siderophore is sort of like an assembly line, and if we can put a drug into the bacterium that acts like a wrench to stop any part of this assembly line machinery, well, then the siderophore will never be made and can never gather iron, thus making the process of getting iron much more difficult for the bacterium. There are some bacterium, however, that are actually very good at keeping antibiotics outside of their cells. And in this case, we could use what we call a Trojan horse method. We take an antibiotic that we know works against this particular kind of bacterium, we link it to the siderophore, and trick the bacterium into bringing in the siderophore with the antibiotic attached. The more siderophore that the bacterium brings in, then the more antibiotic is there also. And we can use this to more efficiently get antibiotics to the bacteria like pathogens. So the next time you think about iron and how important it is for your daily diet, I hope you'll remember our cast of characters. The bacterium that need iron also, and how they make siderophores in order to gather iron from their environment. And then there's siderokalin, our defense against siderophores, binding them so they can't get back to the bacterium. But then also the stealth siderophores that are made by pathogens and avoid siderokalin so that they can take iron back to pathogenic bacterium more easily. And all of this complexity over something as simple as iron. Thanks for listening. If you enjoyed this 10-minute video about the battle for iron, then you might want to check out some of the other cool videos from Relate. You could also follow me on Twitter and learn more about this particular topic or about other cool topics in science. Hope you have a great day.